Hi everyone, and welcome to Biology Professor. Today we're talking about pseudogenes, so let's go ahead and get started. First, what are pseudogenes? Pseudogenes are former genes that have accumulated mutations. And this is important because they've accumulated enough mutations which alter the nucleotide sequence, that alter the gene enough that those mutations make these genes non-functional. So genes in the genome of an organism that are so mutated that they are no longer functional. They're sometimes called vestigial genes, vestigial. What this means is that they have lost their function in the course of evolution. The idea here is that these pseudogenes are descendants of genes that were functional in ancestral organisms. So long ago in the distant past, these were functional genes that carried out important functions for the organism. But over time, over many, many generations, they picked up mutations and eventually became non-functional. And so you can think of these pseudogenes or vestigial genes as being remnants of once functional genes. Now let's talk about the mechanisms of origin. Basically, how do these pseudogenes come into existence? And there's basically three different mechanisms that we know of. One is that it might be a gene whose product is no longer needed for survival. So the idea here is that it was a gene that was once important in an organism, but over time, as the organism's environment changes, as the population of these organisms adapt and encounter different things, there are certain genes whose product, and remember that the product of a gene is a protein, usually, and so those proteins are simply no longer needed for survival. And what's a good example of this? There is a bacterium known as Mycobacterium Leprae, or I'm going to abbreviate it M. leprae, and this is actually the bacterium that causes leprosy. And it's a great example of this because it has over 1,000 of these pseudogenes in its genome. That's approximately 50% of this bacterium's genes that are non-functional, that are so mutated that they don't work anymore. They're not being expressed anymore. And how can this bacterium even survive that? Well, it turns out that Mycobacterium leprae is this obligate intracellular bacterium. It survives and replicates within human cells, where it uses human cell products. So it uses enzymes and other sort of materials and metabolic processes that are present inside the cell. So it no longer needs to make those proteins by itself. And so the genes that originally coded for functional products in a far distant ancestor of Mycobacterium leprae, they just aren't needed anymore. And because they aren't needed, they have accumulated mutations and the bacteria just went on without them. And so that's a good example of that one, where a gene product is just no longer needed. Another mechanism is when a gene is duplicated. So this is when you have a gene in the genome and it gets duplicated. So now you have two copies. And one of those copies, the original or really either one, remains unmutated and thus functional. And as long as you've got one that is remaining unmutated and functional, the other one, the duplicated one, it's free to accumulate mutations and to be non-functional without harming the organism's chance of survival. And so when you have this duplicated gene, where the original remains unmutated and functional, the duplicated gene becomes a pseudogene over time. Great example of this is hemoglobin. So hemoglobin is the protein. It's got 4 subunits. It's in red blood cells. It carries oxygen from the lungs to the tissues, and it carries carbon dioxide from the tissues back to the lungs. Very important protein. And it is made up of 4 subunits, 2 alpha subunits. So one of those is a duplication of the other, and 2 beta subunits, with one being a duplication of the other. And those are all functional. But there are additional duplications beyond those first duplications. So there are, I believe it is 5 more duplications. And those 5 additional duplications, they're just not needed. It doesn't matter if they pick up mutations and become non-functional, because the other alpha and beta genes are fine and are functional and are working in the body. And so those duplicated ones can pick up these mutations. One of them is still being expressed. The others are so mutated that they're not being expressed at all. But that's a great example of this additional mechanism through which pseudogenes can arise. The final mechanism is what we know as a processed pseudogene. A processed pseudogene is a result of a DNA copy of mRNA being inserted into the genome. So remember that through the normal course of genetic information flow in a cell, you have DNA being used as a template to make mRNA. Well, sometimes you can have a situation where the mRNA is used as a template and the DNA is a copy of the mRNA. That's called reverse transcription. So when you have reverse transcription and you make this DNA copy, and that DNA copy then gets integrated back into the genome, that process is called retrotransposition. An example of retrotransposition that adds this non-functional DNA copy of mRNA back into the genome are pseudogenes for both actin. Actin, remember, is a protein subunit that makes up microfilaments that are an important part of the cellular cytoskeleton, and also ferritin. Ferritin is an iron binding protein. Ferritin binds iron to store it inside an organism so that it can be released as it's needed. And so both of these genes for actin and ferritin at some point in the human history were made, copies of mRNA were made, and then some of those copies of mRNA were copied back into DNA, that DNA was put back into the genome, and we have these non-functional pseudogenes of actin and ferritin hanging around in our genetic code. Now, before we leave, I want to point out an important caveat, and that has to do with this word right here, non-functional. For a long time, it was assumed that all pseudogenes, all of these genes that were former genes that had accumulated these mutations, that they were non-functional. And in many cases, that's true. But scientists are beginning to understand that some pseudogenes do appear to have some regulatory function. They might be able to regulate how other genes are being expressed in the genome in a mechanism similar to something called siRNA. If you are interested in learning some more about some of these topics, I have some other videos that might interest you. One is over regulatory RNAs, where we discuss the mechanism of siRNA in more detail. So if you're interested in that, check out that video on regulatory RNAs. Another one that you might look at is my video on the Central Dogma, which we'll go over this idea that DNA is copied into RNA and then protein is made, and you can see how these processed pseudogenes are an exception to that rule. And that is it for today. So thanks for watching Biology Professor, and I hope to see you next time. Bye.